Hello, everyone. I'm David Yoder, Senior Manager of Product Strategy for Aldevron. Thank you for joining me today as we discuss CRISPR ribonucleoproteins, or RNPs. So, the first generation of CAR T therapies were Kymriah from Novartis and Yescarta for Kite. These were approved back in 2017, and they were modified T cells expressing a virally delivered CAR gene against CD19. Since then, additional research has shown that knocking out TCR genes prevents dimerization with exogenous TCR and abolishes cross-reactivity. Further, knocking out PD-1 enhances T cell longevity and activity and reduces T cell exhaustion. And finally, removal of HLA genes eliminates graft versus host disease, which can allow for allogeneic therapies. So this RNP process is a complexation of a CRISPR nuclease, in this case Cas9, and a guide RNA. This complex RNP would then need to be QC validated, documented, and then the RNP is electroporated into mammalian cells in an ex vivo process. Typically, this is done on a case-by-case basis or maybe at a point of care. And so each individual complexation is a new QC event. What Aldevron is offering is an RNP drug substance that we can support you from development through IND filing. So we have multiple steps here, starting with a research grade development where we do a small scale up of the RNPs for optimization and guide resuspension. That is followed by research grade manufacturing, two stages, again, to ensure that scaling up is successful. It also encompasses a force degradation study, a stability study, and a freeze-thaw study. That's then followed by QC validation, where we qualify the reports and verify all of the data against your specific RNP using our QC panel. From there, we move into our GMP manufacturing, beginning with an engineering run and followed with the final GMP manufacturing. And as you can see here, we offer QA reviews, TCE, BCE statements, C of A's, certificates of compliance, and MSRs. The amount and quality required for RNP manufacturing of both Cas9 and guide are given here in generalities. So as we move through each stage, we need more and more material, and the quality of that material shifts. So we can use research ISO 1345 manufactured Cas enzyme from Aldevron for our research grade development, followed by our GMP manufactured Cas enzymes for the engineering and GMP lots. For the guide, we can start with research grade. However, it's best to use HPLC purified research grade material or engineering grade material from development through the research grade manufacturing. Once we get to the engineering run, then at that point, we would need to be using GMP manufactured guide material. For the QC panel assays, we have product characteristics and critical contaminants. For the characteristics, there's concentration by Bradford, there's an in vitro activity cutting assay, an appearance test, residual-free intact guide, residual-free Cas9, pH, and osmolality. For the critical contaminants, we test for residual DNase activity, endotoxins, bioburden at the research grade and sterility at the GMP level, and mycoplasma. For measuring the free Cas9, we use ACEX. So you can see here on the left panel, the unbound ApoCas9 on the left and the bound RNP on the right. So when you overlay these two, we can see that there is a significant difference in the migration between the bound RNP and the unbound ApoCas9. We also measure free guide. On the left, you can see a native page gel, and on the right, a Western blot. We can see that the bands at the top of the gel represent Cas9 protein, and the band at the bottom is our free guide. We also need to consider the impact of the quality of the guide RNA. So on the left, we see a gel with different guides and purities. The more smearing that we see on this native gel, the more likely it is to interfere with our downstream assays. For example, here, looking at bound and unbound Cas9. So on the right side, the bottom panel, you can see there's significant pull-up of the baseline, and this correlates directly with the level of smearing. So this is why HPLC purification is critical for manufacturing RNPs. For long-term stability, we first looked at up to 6 months cutting activity at -80, -20, 4 degrees, and 30 degrees C. We can see at -80 and -20 the very similar cutting activities, and a similar profile at 4C, but we are beginning to lose cutting activity at 30 degrees C. Moving out from there, looking at the stability of the RNP itself, again, we see -80 and -20 have very similar profiles. We do begin to see a little bit more baseline pull up at 4 degrees C, and at 30 degrees C, we can see that the complex is degrading. So for long-term stability, we only looked at -80 storage. And here we see 6 months and 12 months. We see very similar cutting activities and very similar RNP purity profiles. Then comparing 12 months and 24 months, again, at the top gels, we see very similar cutting activities. In the bottom panels, we see very similar profiles for our purity. We also looked at free guide stability. So here we have 3, 6, 12, and 24 months. So we can see that over time, there's very similar stability at all the concentrations at each of these time points. At the 3 month time point, you can see we also measured 4 degrees and 30 degrees C, and you can see complete degradation at 30 degrees and significant degradation at 4 degrees. So what are the benefits of a large-scale RNP manufacturing? You get single lot manufacturing. That gives you reproducibility with a validated QC panel, long-term stability, and IND support for your FDA filing. So thank you for your time. If you have any questions, you can reach me at david.yoder@aldevron.com. Thank you.